panel discussion of the day. We will be happy to take any questions you have. Please write your questions in the Ask a Question tab that is on the right of your screen. And now to introduce the panel and the speakers, I'd like to welcome Mr. Gagan Singh, Country President and Managing Director, Astrazeneca Pharma India Limited. A very warm welcome and over to you, sir.
Thank you, Khyati, today. First of all, I think really excited to be part of the OPPI annual day, and I think the theme and the video was very, very inspiring. We're beginning to see the Indian pharma industry make significant stride towards R&D. Increasingly, the spends on R&D continue to go up for the Indian pharma course. And of course, they want to play a bigger and bigger part in the global R&D space. But without much ado, we've got some fabulous speakers, particularly global speakers, who are going to give us insights in today in new frontiers in vaccines and drug discovery. And I'm really glad to share with you our list of fabulous speakers who've lined up. So a big thank you to all of them for joining today afternoon in India. Beginning with, we've got Dr. Johanna Bendel, who's the Global Head of Pharma Research, Early Development, Oncology, Roche, and would love to hear your views on how we're progressing in, oncology. Dr. Raman Rao, the Chief Executive Officer of Hilleman Laboratories. Dr. Darryl Mcconnell, Senior Vice President, Research Site Ahead, Boehringer-Ingelheim, Regional Center, Vienna, Austria. So thank you so much for joining today. Klaus Urbahns, and I hope I've got your name correct, Klaus. Apologies if I haven't. Global Head of Discovery and Development Technologies, EMD for Serono. And we've also got Inge Lefevre, the VP Head Development Vaccine Business Unit, Takeda. So big thank you for joining here. And moderating our conversation is none other than Dr. Shoibal Mukherjee, who's a clinical pharmacology and drug development expert. Without much ado, Shoibal, over to you.
Good morning, good afternoon, good evening, everybody. It's a pleasure to be here this afternoon with this very interesting panel discussion that we have in front of us. We have, as Gagan just said, a stellar panel here, and we look forward to discussing new frontiers in vaccines and drug discovery. We have on the plus side the fact that because two of the panel members may not be there today, we are able to spend a little more time with our panelists and hear more from them than we had originally thought we would be able to. So with that, this is how I look forward to taking this discussion forward. Let's have two rounds of discussion. The first round would be forward-looking. We'll ask our panelists to talk about new technologies, what excites them, what they think is going to drive research in the years in the near future, and then we will go around once again, asking them what the challenges are, asking them what keeps them awake at nights, what they think that the pharmaceutical industry, the research-based industry particularly, could do to address these challenges, and perhaps what governments and civil society and anybody else for that matter could do to mitigate these challenges to any extent. So with that, I call upon first Dr. Raman Rao. Dr. Raman Rao has been Chief Executive Officer of Hilleman Laboratories here in New Delhi. It's been just over 10 years now that Hilleman Labs has operated from Delhi. They have recently moved headquarters to Singapore, but Dr. Rao has had the experience of leading this organization in the last few years. And it would be very good to hear from him what that experience has been, how that experience is going forward, and what he thinks the future holds in the next 2 to 5 years. So over to you, Dr. Rao.
Thank you. Thank you, Dr. Mukherjee, and hello to all the esteemed members on the panel. I know many of the other members are probably much better placed to talk about drug discovery and those elements. I'd like to focus on a certain elements on the vaccine development value chain, because my core, while being in vaccines and biologics, is that. So I'd like to focus on what you said, Dr. Mukherjee, are some of the trends that evolved over the past decade of existence for Hilleman, but more importantly, globally, in the vaccine and biologics development space. And even then, I'll probably focus on a couple of areas which have become more significant, especially as COVID has shown us and in the evolution of the past couple of years. One is, of course, manufacturing technologies, and the second one is regulatory sciences. I think both of them play a huge role, along with other areas of the development value chain. But I'd like to kind of focus on those and try to give a view on how those have evolved and why I'm excited about certain areas in manufacturing technologies and regulatory sciences. So, I mean, being in vaccines over the past couple of decades with a couple of major MNCs and before getting into Hilleman has given a perspective as to how intense, and then both in terms of CapEx and OPEX, vaccine development and biologics development in general are, and in the time it takes. And there are, of course, several pain points as we can look at it. The early CMC development is challenging, when you need to spend a lot of time and resources. And then as you transit into manufacturing, validation and those elements take a longer duration before commercialization. So there's a huge impact and investment that companies put in there. And all of this is time consuming, which of course, we've seen certain nuances change in these areas that has helped evolve vaccines probably more towards the future for a rapid development. And the most important thing for me is platform technologies. Now, traditionally we have had the standard cell culture based vaccines. We have inactivated vaccines. We've got recombinant proteins. Of course, monoclonals have always been mostly based on CHO cells, but we've also seen an evolution on newer platforms, which could have an impact on, most importantly, the cost of goods. And these can be newer expression systems, which are going to play a significant role, in my view, in the coming future, and especially have an impact on low and middle income countries, and also access. From an access standpoint for biologics, these novel expression systems, whether it's yeast, whether it's calculated yeast or converted yeast systems, or newer mammalian cell engineered systems, which can have a significant impact on the function and evolution of both monoclonals and also vaccines. The other area where I'm pretty excited about, and I've seen rapid development, is what we call as continuous manufacturing technologies, which are modular, which are able which are scalable, and which are technically in a box. Now, whilst we do not have very significant commercial products yet, but we have seen a pivot even within large pharma to adopt these technologies that can potentially make manufacturing and validation within manufacturing a little bit more smoother. And so these continuous manufacturing technologies, especially for vaccines, biologics, can play a huge role in having an impact on development, impact on commercialization, the timelines, having a positive impact on the supply chain, of course, and then cost of goods, and thereby having a positive NPV on each and every one of those development programs. The other element I'd also like to allude here is, when we talk about manufacturing platforms, digitalization of certain sectors of manufacturing has also evolved. And this is more towards the downstream, what we call as the drug product area, the packaging, labeling, and those areas, which have had a significant impact on timelines. And especially after your quality control, after your quality checks, the product out of your facility, those times have actually had a positive impact as we've used digital technologies in the downstream areas. I do feel that the digital technologies will take a little bit more time from an adoptive standpoint upstream, but downstream, there is already a significant impact on evolution of products, the release and the supply chains, which utilization of digital technologies. So these are more the larger scale examples where I've seen that I'm pretty excited about on the manufacturing side. There is also a little bit of an early adopter mechanisms where when I mentioned CMC, which has significant pain points, especially the analytics, we have seen newer technologies and newer platform technologies with multiplexers, octets, and those mechanisms come into the analytical development area, which will have a significant impact on the development of biologics and vaccines, which I'm very proud to have used before. Worked with teams who have extreme amount of knowledge in that, but also they will have moving forward an impact furthermore as we evolve the product more into validation. So those are some of the examples. Having seen those in live, I do feel very excited about, and more the adaptation of these will have a positive impact on the development value chain. There is one more area which I call as platforms, which have evolved as well, and which COVID has shown to have had a positive impact, which are adjuvants, the use of adjuvants. We've for long spoken about the value of adjuvants. We've had only one primarily, which is alum, different varieties of alum, which has been used in different modalities over the past 50 years. We've been able to add only a couple more novel adjuvants, with the GSK is primarily taking the lead in that. And of course, Novartis has their MF59. So these are some classical examples of adjuvants which have taken a long time to evolve. With COVID and with the emergency utilization mechanisms in place or instituted, we've seen quite a plethora of adjuvants being used clinically in even regulatory arenas where they were significantly risk averse to use novel adjuvants. So that's one platform which I do think will have a very significant impact moving forward, and which I'm fairly excited about. And finally, just one more element that I want to allude to out here is the overall regulatory framework and the regulatory sciences arena, which has also evolved. And more positively, especially as we've seen with COVID, where there is a positive dialogue happening with the regulators early on in terms of adaptation, in terms of novel mechanisms, in terms of instituting a novel program where your product can go through development and approval. And we've seen rapid timelines. Of course, I mean, they're emergency users, but still they help evolve the program faster. I mean, these are timelines which I've never seen in my life before, especially with the mRNA platforms and other adenovirus vector platforms. We've seen that happen with the monoclonal antibodies as well. So regulatory sciences has also evolved within these 2 years. And I do believe that in a positive sense, this will have impact in overall development framework as the industry move forward in developing novel solutions. So those were my thoughts as I look forward to certain elements which I'm very much excited about. I'm pretty excited as we utilize them more towards developing novel vaccines and biologics. Over to you.
Dr. Rao, a quick follow-up question on that. So, you've talked about platform technologies and continuous manufacturing technologies and stuff like that. And there's no doubt that, these are going to enhance or smoothen manufacturing and help with efficiencies of manufacturing. Now, how does that translate, just for our audience here, how does that translate to the patient? What does the patient get out of this? And how is the patient's life improved? Does that impact access, and how does it do so?
There are two major ways this can impact. One is because your development timelines could potentially have a positive upside, the reach to the patient is faster, number one. Second, if you're able to make more product within a certain time limit with modular manufacturing, your access in terms of ability to provide the more number of doses to the patients in the end also improves. And access has always been a crucial area for consideration for all the pharmaceutical industry, because at the end of the day, you have your cycle times, and those cycle times are traditionally fixed based on your manufacturing cycles. If you're able to reduce those, which will then also have your indirect impact on your cost of goods, thereby allowing you to provide patients with more, probably cost-effective medications and also providing a larger access pool to the patients. So it's positive on those fronts.
Great. Yeah, we do hope that that will lead up to better access of vaccines. Particularly, that is one area where access is indeed a key and critical factor. You also talked about the regulatory aspect. So I now want to go on to Dr. Inge Lefevre, who is VP Head Development Vaccine Business of Takeda, the question I'd put to her is, Dr. Rao talked about regulatory aspects, and now we're seeing, a globalization of the pharmaceutical industry. We see, goods produced in one country being widely exported, and I'm talking here particularly of India, where we are hosting this conference, exporting to almost all over the world, and yet. While there has been this movement towards harmonization of regulations, how far has it gone? How positive are you that it is going to further get on to a place where regulations across the world and differences in regulations across the world are not going to create hurdles in taking products from one location to the other, manufacturing in one place, providing succor to patients in another location?
Thank you for the question, and thanks. Hello, everyone. And I was indeed hoping that my part of the talk would be complementary to what Dr. Raman is saying on the manufacturing side, and also speak a little bit about the R&D side for vaccines, because I do think we also see some general trends there over the last years. But coming back to the regulatory and expediting regulatory reviews, I think we've seen a tremendous opening with the COVID vaccines in terms of agencies accelerating their review. That has not necessarily led to a harmonized review, but I do think that moving forward, there are a lot of initiatives coming out of a certain agency to streamline and really look for opportunities on how we can better harmonize our review or their review and come to a more global assessment of the quality and efficacy and the safety of the product. If I can just give you an example of what we're trying to do. So Takeda has moved from a Japanese vaccine manufacturing to a global vaccine developer, and we're quite advanced with our dengue vaccine, where actually we're trying a new mechanism as well, because we want to make the vaccine available first in the endemic countries. And so we have EMA actually has accepted not only our European application, but also under the Medicines for All, which means that the same 6 or 8 countries, endemic countries, are part of that review procedure. And so they can share their reviews, they can share their concerns from their local requirements and try to come with a more harmonized, first of all, accelerate the review, but also come to a more harmonized way of reviewing by sharing the questions. And we see that the individual countries that are also reviewing the file at the same time, they can share their concerns and they can share their questions. So I do think that type of mechanism can help in getting to a more global approach to medicines and vaccine approval. On the other hand, I do still think that work needs to be done on how the review process can still be expedited, in the sense that vaccine development, as Dr. Raman has explained on the CMC side, is very complex, but it is also very complex and very heavy on the clinical and safety side. It requires a huge data set. It has a very low threshold for safety. And that's most of the time the concern, especially with vaccines, is the safety of the vaccine. And so there is a lot of research ongoing to see how can we get quicker access to the data, new ways of getting to these data to provide earlier an assessment of the safety instead of waiting for the long time until the full data package is actually completed. We've seen it under the emergency use with the COVID vaccines. We should not expect every product is going to have that same status, but I do think it has opened the door for more research in finding alternative pathways to registration. It has always been there, a little bit in the background, but I think now we can see a movement towards that. And the same thing with the countries being involved in these reviews, looking at subscribing to the ICH, standards and so forth, that will also help the countries to have a more standardized review for new products. That's what I would add on the regulatory side. I just wanted to bring out two more points, which I think we have seen not only in vaccines, I think in general in the R&D space, is that in recent years, there's an exponential growth in collaborations and partnerships between not only agencies and companies, but between companies, but also companies with research institute, which could also accelerate and in finding new technologies and accelerate research and development. And the second thing, the third thing then was also brought by Dr. Raman, is that, yes, the investment, especially if you think about vaccines, what we've seen in COVID is it requires a huge upfront investment. And I think companies, countries, institutions can only do what they can. And finding alternative pathways of funding research and development, I think, is also something that we see more and more. In order for everyone in the development field to find new technologies to accelerate, moving from non-clinical to clinical, finding new areas and new pathways of funding, I think, is also something that we see more and more. So those were my 3 points that I wanted to contribute to Dr. Raman's statement.
Great, thank you so much, Dr. Inge. Just to follow up here, just out of curiosity, that you talked about these 8 countries that collaborated in the regulatory review of this dengue product. Now, was India part of that? After all, India is an endemic dengue country.
Yes, no, absolutely. No. We are currently in the process of starting the development in India. So no, it was not part of the initial clinical data package, but moving forward, obviously, we will also explore that possibility. Yes.
Great, thank you so much. It was so interesting to listen to this. I will go on now to Dr. Klaus. Dr. Klaus Urbahns is Global Head of Discovery and Development Technologies at EMD Serono, based in Boston, I understand. My question to you, Klaus, is what new technologies are out there that are exciting, that you think will change the way that we do research in our laboratories in the next kind of 2 to 5 years, and how is that change going to impact researchers, research itself, and patients at the end of the day?
Oh, thank you so much. I think it's fair to say that there's a lot happening at the moment. We just touched on the messenger RNA vaccines, but I believe that the RNA as a molecule more generally will play a dominant role in the future. I think we will see more medications like short interfering RNA and other things. Even antisense oligonucleotides are having a huge comeback, I think. So this is something more generally where I would see this is a very interesting new development. Obviously, we've already spoken about cellular therapies. That is yet another area, whether it's these famous CAR T cells or natural killer cells or what have you. I think what we are seeing there is maybe a slight bend in the curve, because I think increasingly regulators are realizing that this is a kind of also something that needs to be monitored more closely from a safety point of view. But still, of course, this has, of course, a huge potential. These are probably the most popular modalities where you really see a lot of progress, and which will make a difference, because all of a sudden certain diseases we will be able to treat certain diseases in a way we never could before. But I would still also refer to the more traditional modalities that we know of. One is clearly antibodies. I think clearly we are seeing a move away from the traditional antibodies towards, I think, multi-specific antibodies, which are way more complex in terms of making them, in terms of monitoring the CMC aspect. It's always a difficulty actually in creating them the right way, so that both arms or three arms are really having the right ratio towards each other, so that in the human body where they freely float around, they really get the right proportion of their target and the target occupancy everywhere. This is a challenge, but I think it's a fantastic opportunity as well, because we can really do, if for instance two receptors on the same cell, we can really expect synergies in many cases if we see that we can bring two cells together, for instance, through an antibody that sits in between. That is a fantastic opportunity. These kind of engages. So there's a lot actually that can happen. And finally, and last but not least, I would really like to get back to the most classical small molecule area. I think that we see a lot happening there as well. It's not only that we see increasingly formerly non-druggable targets being drugged all of a sudden because people are becoming more inventive, but also what we see is this new way of actually engaging with a receptor, which is not only to inhibit it pharmacologically or modulating it agonistically, but actually taking the whole protein out of the system. So these new degrader molecules called PROTACs. This is in many ways the dream of any molecular biologist. If you think about how targets are being discovered and validated today, it's basically by taking them out of a cellular context. And then we look into what pharmacology results. And in the past, we have always tried to mimic this by actually modulating and inhibiting this, and always had the equation in the back of our minds: taking something completely out is the same than inhibiting it. And often we were confronted with unpleasant surprises that it is not the same. And now I think for the first time, we can really mimic this and really take the target out. And that is a completely new thing, I believe. We will become better in translation. I think with these kind of molecules, we will be better in predicting things. We will be able to mimic knockout situations with these molecules in a pharmacological way, such that we can dose them. The newest generation of these molecules are also oral medications. So although despite very large molecular weights, we are still able to understand the tricks to make them actually orally available for our patients. So convenience will be there, along with new therapeutic paradigms and completely new ways of treating disease, I believe. So on a modality front, I see lots of progress everywhere, and that really makes me optimistic for the future.
Great. So nice to hear, and really interesting topic of PROTACs, which I'm not going to ask you more about because I want to reserve that for Dr. Darren Mcconnell, who's recently published some stuff on this. But before I go away from you, I want to ask you, Klaus. We've heard of, and of course we know the way that these two new vaccines for COVID work, the mRNA vaccines. Now, immunotherapy for cancer. How far are we from seeing an mRNA vaccine against cancer, or where is that going, and where do we stand on that?
Very good point. I mean, I think we have to admit that so far the world of vaccines and oncology has been a little bit frustrating, to put it mildly. I think there were many setbacks, but of course there is now a lot of hope that with the new modality of messenger RNA-based vaccines, things might change. Also, I think the clinicians or the clinics has actually changed. I think we are getting smarter in choosing our approaches. One example, for instance, is to go increasingly early in selecting those indications where that is possible. One of those areas, for instance, is colorectal cancer, where many of us are getting coloscopy, and if you're at that age, actually, where you're actually being investigated whether there's any polyps or anything growing there that shouldn't grow. And that is an opportunity, looking very, very early before actually the tumors start. And those indications, I believe, when we can interfere early, we will be able actually to also apply those vaccines more successfully than we did in the past, along, of course, with messenger RNA vaccines, which seems all the way to be more powerful. We have, our neighbors I probably should explain that EMD Serono is actually part of a larger German company called Merck in Darmstadt. And Darmstadt is very close to the city of Mainz. And Mainz is actually the home of BioNTech, where the first messenger RNA vaccine. And they have a fantastic pipeline, our neighbors there, in terms of oncology. It's multi-targeted vaccines, and they have really encouraging first clinical results. So it seems that there is life in that area again, and I'm very optimistic that things might pan out, and I really hope the best for them and for patients, that this might really be successful.
Great, thank you so much for those comments. We'll move on to Dr. Darren Mcconnell. He's Senior Vice President, Research Site Head at Boehringer-Ingelheim, based in Vienna. And Darryl, you've talked at length. Tell us a little bit, particularly the products and what they do and how they can be applied to different areas of disease.
Dr. Mukherjee, yeah, I'd love to. Can I just first say, I almost feel like I'm in India. I haven't come to visit you all for many, many years, far too long. I love the country, I love the people. And it's wonderful to be speaking here. And I certainly share Klaus's excitement about all the modalities. And I'm really, really glad, Klaus, that you mentioned the classical organic chemistry. I'm an organic chemist by training. I haven't been in the lab for a while. I know there's many, many, many talented chemists, organic chemists in India, some of whom we collaborate a lot with. So I think it's a great topic. And this PROTAC, new technology of PROTACs really has, I think, for the sort of small molecule area, it's the first big sort of innovation we've seen in the field of small molecules for quite some time. And what a area of innovation. It really opens up so many opportunities. It's not just degradation, right? There's new.
You've got to explain this a little bit, Darryl, to the audience. They need to understand how it works. Just in a few sentences.
Everyone should know it by now.
We have a diverse audience here, so some of them might not really understand the technology so quickly.
No, I joke. So these are molecules that are bispecific molecules. So similar to, we have T-cell engagers in the antibody field. These bispecific monoclonal antibodies that Klaus was talking about, in the antibody field, they glue a cancer cell. So they recognize, a spike protein on the surface of a cancer cell with a spike protein on the surface of a T cell, our killer T cells, and bring them together and kill the cancer cells. The PROTACs are the same, but they're made of small molecules, not big proteins. So you've got sort of two small molecules on either end, and they are designed to hijack things inside the cell. So they actually get inside the cell, they bind to your protein of interest. Let's take my favorite protein, KRAS. This is the beating heart of cancer. We've been trying to drug it for 40 years now. They bind to KRAS, and at the same time, they bind to other molecules in our cells that are called E3 ligases. And these are essentially the sort of used by date proteins in our cell. They stamp proteins with a you're out of date signal, just like your milk and your use by date in the fridge, to say that protein is out of date and should be shredded by the so-called proteasome, the garbage disposal system of our cells. So we can, as Klaus nicely put it, we can not just turn these disease-causing proteins off now. We can shred them completely from the cells, and we've and others, of course, we've been got into this technology early, and we've been applying it in particular to KRAS, and we were able to massively degrade KRAS in vivo in mice and shrink tumors of all, essentially for all the different KRAS mutations. So it's a very, very powerful modality. And
Darryl, does this technology then extend to say degenerative diseases? Can it be used against those as well?
It's being applied beyond oncology for sure, and many areas actually. Yeah, oncology is as it often is. I'm sure Johanna will talk about this further. You know, the need is so huge, and oncology is often for new technologies, new modalities, new regulatory approaches. Also, you know, the sort of spearhead. But immunology diseases, yeah, many areas it's being applied to. Yes. Yeah, I actually said when Protax first came out, I said, yeah, Protax can be applied to everything except to CNS diseases. But it's being applied to CNS diseases as well.
How did that happen, and why did people say that it cannot be applied to CNS disease?
Well, I just that was me, and you know, usually I'm the one who says everything's possible. I mean, getting through the blood-brain barrier. So we have around our brain, we have essentially a fortress of sort of vasculature that's designed to you know, spit out any molecules that it doesn't like. So it's actually very difficult, even for classical small molecules, to get them into the brain. One of the challenges in oncology, of course, is brain metastases, very frequent in lung cancer.
Is there something special having to be done to get these molecules, to get these proteins into the across the blood-brain barrier?
I don't know. We're not sort of at the forefront of that. We're sort of more pushing the oncology Protac field. But Arvinas in Connecticut in the US have got, amongst others, but they have a lot of efforts in that area and have had some successes in mice and getting Protacs into the brain. So you probably have to ask them what their tricks are.
Okay, so we move on to Dr. Johanna Bendel. Dr. Johanna is Global Head of Pharma Research and Early Development at Oncology at Roche. And from an organic chemist, now we come to a hardcore clinical oncologist. Dr. Bendel, tell us what excites you in the next few years. What do you think is going to drive research in your area and in your company?
Well, thank you so much, first of all, for allowing me to be here with this esteemed panel. I think that we've touched on a lot of things already, so maybe I'll even start to group them together. I think within cancer therapies, we're seeing the advent now that we've seen immunotherapies work in cancer patients, both in solid tumor malignancies as well as hematologic malignancies, almost a renaissance of development within the immunotherapy world. And we've already heard about some of the big approaches that we're taking, from where we started with checkpoint inhibitors to next generation checkpoint inhibitors. We just saw an approval this past week by the FDA of LAG-3 inhibitor in combination with PD-1 inhibitor for patients with first-line metastatic melanoma, which is sort of a next transition into combinations of next generation checkpoint inhibitors. We're also seeing the bispecific antibodies, as we spoke about before. How can you put on one molecule be able to target two things or even three things potentially to create an immune response? And some thoughts are there are even bispecific PD-1 LAG-3 molecules that are out there that, as opposed to two separate antibodies being given at the same time, if you have the molecules together on one and you tightly bind to PD-1, could you more target that therapy towards the PD-1 expressing cells than just giving the two antibodies together? So the bispecific technology has a lot of potentially interesting approaches to it. We also heard a little bit about the bispecific technologies bringing T cells targeted to the cancer. And we've seen in hematologic malignancies primarily effect there where it's almost close to what we see with the CAR T therapies. And now we're seeing that we can create bispecific antibodies and give them in combination with co-stimulatory molecules, where you could almost mimic a second generation CAR T, where you bring the T cells to the tumor and then you add on a co-stimulator at the same time. So there's a discussion now about cellular therapies and bispecific therapies, and can one be just as good as the other? And does CAR T therapy, for instance, you have a very intensive therapy for a short period of time, but then you don't really need as much follow-up therapy, where, for instance, bispecific molecules would be therapies that you would have to be given more chronically, but you wouldn't have that short-term intensive therapy. And so there's a lot of discussion going on right now about how to optimize these approaches for patients. And certainly, with the cellular therapies, as we were talking about right now, a lot of the cellular therapies, you require the patients to have leukapheresis and remove their cells, engineer them, and then put them back in again. But what the next generation is the off-the-shelf cellular therapies that will come in, where you have a donor T cell, which is able to then create that same effect without needing to take the patient's own cells out. And so how will that affect the next generation of cellular therapy design? As well as the cellular therapies, when we talk about vaccine therapies, and it's just so interesting with this, another renaissance that's happening within the mRNA vaccine world. There's also more vaccines aside from mRNA vaccines that are being looked at in the cancer world, including dendritic cell vaccines, where we've already got an approval for prostate cancer within the United States. But how can we look at vaccines? There's technology using antigen presenting cells and creating an off-the-shelf vaccine approach with these antigen presenting cells and oncolytic viruses that are coming through, and how do we start to optimize those? But what I find very interesting is when we talk about off the shelf, right? So you can just give this to a patient right away. When we talk about something like the BioNTech mRNA approaches, they're off the shelf approaches, but they also have personalized cancer vaccines that they're developing. Moderna is also doing this as well, where you remove the patient's tumor, you create a panel of antigens that you then put back a cassette that you put back into the vaccine and give it to the patient. And then can you create a personalized tumor vaccine for those patients? And so that's sort of the next thing that we think about coming along is that personalized treatment of a patient's cancer. And I really appreciate the discussion of the immunotherapies as well. Where is the best place to use them? Because we know that as opposed to traditional chemotherapies where we can see tumor shrinkages in a few weeks, the immunotherapies actually need a chance to stimulate the patient's immune system, create an immune response, and then have an anti-tumor effect. So if you have a patient who has very advanced disease, does the immunotherapy have enough time to work? And so could we consider that immunotherapies should potentially move earlier in the disease setting, even into the adjuvant space, where we've already seen now data that shows that in the adjuvant and neoadjuvant approaches with immunotherapies, potentially curative effect of these immunotherapies. And so when we're looking at some of the vaccine therapies, both off the shelf as well as personalized, one place that people are starting to look now is in the adjuvant setting. Particularly, and I also love the reference to colorectal cancer, we now can identify a group of patients with colorectal cancer who have had their adjuvant therapy, but are at high risk of recurrence because we have a blood test to check for ctDNA presence. And people who have high levels of ctDNA after their therapy have a very high risk of recurrence. And so is that the patient population where we should be looking at using cancer vaccines? So much going on within the immunotherapy field. And then when we also talk about large molecules, we're not developing as many chemotherapies as we used to develop, but now we actually have a way to target chemotherapies to the cancers. And we've seen wonderful results with these antibody drug conjugates that are coming, where these next generation, where we have different types of linkers, potentially different binding to the target, where we can successfully deliver a targeted chemotherapy and actually really improve patients' outcomes with the antibody drug conjugates. And then moving.
So out of all of these, Johanna, sorry, did I cut you off? Out of all of these, which one do you think is the one that is going to transform either cancer therapy or therapy in any other domain? Because some of these, like the personalized medicine thing, I don't know how scalable this might be and how quickly this can get to patients across the world. Some centers may be able to do it, but operationalizing it across the world at scale might be a different story.
Yeah. So looking into the very long term, this is what I'm hoping will happen in the future. Is we're also seeing the development now of blood-based testing for cancer screening. So I envision with time, with years, as these screening approaches become validated and become more mainstream, we're actually going to see the cancer patients shift into a much earlier state of diagnosis, where I think we're going to be seeing more patients who are diagnosed and treated in the early stage, where we have a chance for things like vaccines, for shared antigens to be able to work. So my ideal dream is that we are diagnosing patients earlier and we're able to use an off-the-shelf vaccine approach or immunotherapy approach to create a curative therapy for those patients in that precious window where their disease has been mostly removed, but we still have a chance to get the immune system to attack whatever might have been left behind. That's my dream.
Great. Very, very interesting. Let's go on to we've just crossed the halfway mark. So let's go on to the next round of discussions, and we'll probably go in the same sequence. Of course, people have you're free to jump in if you want to make a point. But I'll ask Dr. Raman Rao again. So coming back to you, Dr. Raman Rao, so in all of this, in your experience with Hilleman now and looking forward, what are the key challenges that you see in getting new vaccines to patients? What can industry do or governments do? Is there some kind of collaboration that you would recommend where things could be speeded up and access improved in some way? What's stopping us?
It's a good question, and we all probably at different levels nestle with that. And it's a complex situation as well. When we talk about vaccine development or product development per se, there's a start and there's an end goal. The end goal is to reach the patients, but also during the course, you are able to make an effective therapy or an effective vaccine. And that means risk, right? And that means institutions, organizations, everybody takes risks. And it's how do you modulate that risk and how are you able to modulate that risk-taking capability is going to be important. And that is a multi-pronged approach as well. I mean, that needs investment, that needs time, that needs investment not just by a single entity. And Inge mentioned about collaboration, and I think that becomes extremely crucial. Where you're able to have a balanced approach, that you're able to offset risk and the right investment, you will be successful. So at the end of the day, that means that we'll have to have the right level of investment by all stakeholders, which means it's governments, it's public health authorities, it's the pharmaceutical companies, the academic institutes, and all of those, and sharing that risk across the board to be able to kind of come out with the right solutions. I mean, different countries also have different approaches that they're taking. I mean, one of the challenges I do see, for example, in India, is that we've got great manufacturing, we've got great scale in manufacturing, but some of the challenges are about investment in early R&D, which is probably because of a potential risk-averse nature of companies, and also probably just the way the institutions have been formed over the years. So I think that would then need the governments to actually help forge investments into those early R&D, whether it's through academia or collaborative approaches between industry and academia. And that will then enhance the ability of organizations to take the risk and to fail. I mean, R&D is about failure as much as it is about success. So you won't succeed unless you actually fail and learn from it, and then use that learning into what you're taking forward. So it's a combination of factors. But at the end of the day, it has to be a balanced approach between all these stakeholders and the right level of investment that needs to be made also by the governments, and for example, in India.
So, Dr. Rao, you mentioned industry academia collaboration, and we have a question from the audience. How much industry academia collaboration are you seeing in India, particularly, and are you happy with that situation? What can be done to change? If I'm already presuming your answer, to enhance such collaboration in future.
Yeah, at this moment, I mean, based on the past experience, it does seem that the industry and academia are a little bit in their own silos or their own comfort zones. I mean, academia is probably very great at doing the early science, but do not envisage what are the steps that are needed to take it to a product level. I think that translational ability needs to be bridged, and that is where the collaboration between academia and industry is extremely crucial. And there has to be mechanisms, forums, or even induced mechanisms to bring that forth. So my view is.
Let me put it this way. If Dr. Vinod Paul were to have been here today, what would you recommend to him that he takes with him to the Niti Aayog? And what policy changes would you look forward from the government to make sure that industry and academia collaborate in this country and get somewhere with that?
There has to be a concrete level of investment. It's just that's not monetary, but also in terms of infrastructure that can provide the modalities for academia and industry to collaborate. At this moment, it's still probably in infancy, in my view, based on what I've seen. Vis-a-vis other countries where probably it's a little bit more evolved and there is a structural basis to these collaborations. And so the government really would need to kind of take a look at forging these mechanisms to help evolve this translational piece, because that's at the end of the day is crucial for taking an idea into a product.
Great, thank you so much for that, for those words. Inge, I come back to you. What challenges do you see, and if you could touch upon, what kind of industry academia collaboration you see in other parts of the world, particularly in the part of the world in Europe where you use it, and your words on those.
Yeah, so if I think about in the vaccine world where you want to make the product available for everyone because it's prophylactic, you want to protect people from getting ill. The first thing that comes to mind, and I think Dr. Raman has referred to that, is the continuity of supply. I think that keeps everyone awake in vaccines in terms of how do we plan and secure, because as I mentioned before as well, the production process is often very complex, and so something might go wrong during the production process, which then actually gives an interruption of supply. But there can also be other things like your source ingredients, that there's a shortage, or that. So continuity of supply obviously is very important in vaccines, because the moment you can create a public health problem very quickly if you fall out of supply. If you then think about the development of vaccines, what I still think there is a challenge is also already mentioned, is the risk with regards to the safety of the product. I think over history we've had a number of occasions where even with an extensive clinical development plan that was even excluding risk, if I think about Rota, but even so, when then the vaccine is put on the market and massively used, there is always a signal evaluation that is emerging. But however, that does not jeopardize, the benefit of the vaccine is demonstrated by that time. Therefore, the benefit risk profile still remains highly positive. And so that is still a challenge in terms of when you register a vaccine, how much safety is enough to exclude a risk. One can never exclude a risk. And so usually vaccine files, as you've seen as well with COVID, we have, clinical development plan of thousands and thousands and thousands of people, right? 10,000, 30,000 is not an exception. Mm-hmm. So that's a huge data set. And at some point, I think there has to be some acceptance, and you cannot run your trials in every single country of the world. So you try to select your countries and your subjects in the areas where the endemicity is there, where the population is right, and that you can extrapolate as much as possible to the global population. So it is impossible to target every single country in your development plan for a first license. And that sometimes is as well a challenge, that I think there should be openness to look at the full complete data set as a whole and step away from some specific requirements that, if you have such a large data set where you've demonstrated efficacy effectively, and you have shown that there is no safety issue at that moment in time, that it's safe to move forward from a registration point of view. And the final twist is more about research. I think one of the challenges is that some of these global infections, as we've seen with COVID, but also in other areas, if you think about vector-borne diseases, these are often very complex diseases with a mosquito, with a complex cycle in the human body. And so it is very challenging still and I hope that moving forward, yes, we can use new technologies, new platforms, but also artificial intelligence and things like that to better target new targets for vaccine development, because I still think that is still a challenge that sometimes slow down the development of new vaccines.
Great. Yeah. Thank you. Thank you so much for those thoughts. Klaus, I'd like to change this. Challenges are fine, but there are some pressing things that we need to look into in the near future, and probably get these technologies to address those. For example, what about antimicrobials, and where are we on antimicrobials? Can some of these technologies that we have talked about can they be applied to antimicrobials? Is anybody doing that? And how do we get people to be interested there? And the investment, we've heard a lot about, you need to invest, but in some disease areas, there is not so much pull for investment. So how does one get around that? You're on mute, Klaus.
Sorry about that. It should be better now. It's an excellent question, and I agree. The antimicrobials has been notoriously difficult to find new lead structures, to find new therapeutics. I would wish that we would be in a position soon where we can apply some of these new technologies that we just spoke about actually in the area of antibiotics and antimicrobials. I think that will be a fantastic new thing. I mean, small molecules. Unfortunately, also many of the technologies that we saw in the small molecule field didn't really cut it if it came to antimicrobials. High throughput screening, for instance, was not an area where it was really very successful to deliver new antibiotics, I think. So this has been a difficulty. I think we are kind of mostly, I think, we are drawing from the pool of natural products, which is also increasingly difficult to find new things there. So I agree with you. It's not very easy and frustrating. But on the other hand, it's of course there is an immense medical need. Should there ever be a superbug combining the resistance mechanisms of vancomycin-resistant enterococci with methicillin resistance, Staphylococcus aureus, then this would be worse than COVID. I can tell you. And so let's just hope that bug never exists. But even for those scenarios, we should be prepared, and this could become very, very difficult indeed. So I agree with you. This is an area where we need to invest more. Where I think it's probably something. I mean, most companies have kind of withdrawn from that because it is so difficult. It is probably something where we need to work together with academia more, with private public partnerships, really, where we can actually make some inroads. But at the moment, there is not too much science happening in this field, unfortunately. Again, hopefully something changing with these new modalities that we just spoke about. Antibodies, for instance, could be very, very interesting. Why not having an antibody that has a radiotherapy attached to it and really bind selectively to antibiotics, to bugs? That would be something. It could be very interesting and very specific. So things like that, but you don't see much of it.
Yeah, and companies that had worked in the areas of septicemia and microbial infections seem to have moved away from that area. And I think the only hope now looks like some of these foundations perhaps would put some money there, and we can get somewhere with antimicrobials. But that's something that I think is very scary. So Darryl, your thoughts. What's what are the hurdles in front of you, and how could those hurdles be overcome?
Yeah, well, I mean, we're sort of talking about how to innovate in general, right? I mean, your first questions were around, how could India innovate more? What does it need? And you talked about the link between academia and industry. It's the same question for everyone, I think, whether it be a country or a city or a big pharmaceutical company or a biotech company. That's the question that keeps me awake during the day and keeps me awake during the night. How because the problem is, as soon as you innovate, you've got to innovate again. It never stops. That's the definition of innovation. And I mean, if I can just add perhaps something that might be worthwhile thinking about for India in general. I think that you talk about academia. I think we should talk a lot more about biotech. I mean, I think the two, in terms of life science innovation, and we've talked about, which I'm really happy about, also the manufacturing aspect. I mean, manufacturing oncolytic viruses that Johanna was talking about. There's not many people on the planet who can do that at the moment. So it's taking it right from idea to the bench side. It's those are the two big elements for me: big pharma and biotech. And the third bit is the environment. And I like to talk about hotspots. And this is where the US is just fantastic at it. I mean, Boston. If you want to study innovation ecosystem, it's Boston. And countries often, I speak for my own, Australia, often historically struggle a little bit and Austria actually, my inherited home here, countries often struggle to make a decision, let's make a hotspot in one spot. So get that critical mass of universities, highly educated people, infrastructure, people, biotech, big pharma. Takes quite a while to build that up. And what usually happens when I observe it is countries sort of dilute it and then say, well, we want to, give a bit of money to this part of the country, a little bit to that, a little bit to that, a little bit to that. So you end up with lots of warm spots, and that's not good enough. But once you get one hotspot, as you see in the US, once you get one hotspot, then you can start to get other ones. And it's that sort of entrepreneurial, the sort of biotech spirit and venture capital, which is often, certainly in European countries, the sort of bit that's the bits that it's missing. So anyway, there's just a few sort of random thoughts that.
Right, and it's often a university or an educational institution that forms the the center, the hub of that enlarging circle of innovation in a particular location. In Boston, it's the Harvard University. You go to the west coast of the US, and you have the Stanfords and the UCLAs, I suppose.
But I would argue that it's the critical mass of institutions, universities, and institutes, and highly educated people in the life science centers. That's the key factor, and not Harvard professor XYZ who came up with this idea and started a biotech company. I think that's just secondary. And we're trying to do that in Vienna as well. We have this. We have the wonderful city. Everyone loves to come here, and we've got lots of institutes, and it's easy to get to and well positioned. So we're trying to get that critical mass. And I would argue for us also here, it's that sort of entrepreneurial venture capital investment and lab space for companies and people with cool ideas to tinker and innovate.
Yeah, thanks so much for bringing that point up, and I hope that thought goes around more into policy making in India. Johanna, back to you. And what are your thoughts on the challenges we see in the future, and how do we overcome them?
Yeah, I think I might take us in a slightly different direction, because one of the things that we wrestle with a lot at Roche is the care of what we call the global patient. We have a lot of drug development that's focused around very complicated molecules and complicated treatments that in a Western country is very easy to give, right? Where you could hospitalize somebody overnight, you can treat cytokine release syndrome, people can come back and forth to clinic multiple days in the week to be treated or to be observed. But how do we, and this isn't just an issue with the rest of the world, it's with the West also, and COVID brought this to light, is how do we create treatments that are more patient-centric, where they're not required to come into clinic so often, where they can be monitored from afar with telemedicine? What are treatments that they could potentially self-administer? And so we're lucky with small molecules and some of those treatments where the patients could take the treatment home and then we could follow up with them. But those small molecules have their toxicity profiles as well. And how do we educate patients? For some of these bispecifics, we've had a big focus now within Roche, and I know other biotechs as well, and pharma as well, to say, okay, can we give subcutaneous dosing? And is there a way that with that subcutaneous dosing, we could create, number one, a better safety profile, but number two, a more convenient mode of administration? And so as we think through what those next steps would be, aside from innovation and thinking of curative therapies and further development of our technologies, really pushing towards the concept of making sure we have treatments that can be given globally. And that could potentially be accessible globally, and how do we make that easier? So that's another thing that keeps me up in the middle of the night.
Right. So I was wondering whether anybody has any ideas on the use of technology. So one thing that has changed over the last decade or so is this enormous now penetration of technology into the population. So you look at a country like India, where we are not the richest of countries, and yet almost everybody in the country has a mobile phone, and many of them are smartphones now, and they have their capabilities. Is there a way to leverage that to get healthcare to small pockets across the world in developing countries? That's one thought that I had, and the other is, of course, very different from that. As far as clinical trials are concerned, and clinical trials form 60% of the effort, or maybe more of the effort that goes into getting a new drug to the market. And that's always been, a big challenge, getting patients into your clinical trials. And I'd like to hear from any of you who would care to respond on decentralized clinical trials, and that's a big buzzword among those who work in the area of clinical trials. What do you think of that, and how is that progressing? And do you see that kind of becoming a universal thing in the future? So, question open to anybody who would like to answer those. Any hands up there?
I'll jump in.
Johanna? Yeah. Sure. I was looking towards you.
Yeah, we have a lot of development of apps for the phones, both from clinical treatment standpoints and patient monitoring standpoints, where patients can log in their toxicities, have access to medical advice and nurses, to apps for clinical trials, where we help say, alarm, here's your next appointment, you're due here then, and to help them with and also with medicine taking, to record adverse events. So there's a lot going on within application technology for the cell phones. And I'm glad you brought that up, because that's another way that we could potentially start to monitor patients from afar. The other thing is there's been a big boom in wearable technology. So even in clinical trials and to monitor patient health, not just oncology patient health, where people are wearing Fitbits or Apple Watches that can record health data that then can be uploaded into a centralized repository to help with treatment decisions, to help with medical management of the patient. So I think that we're just almost in the infancy, so to speak, of where some of these technologies can go. And I think a very good point to use what people have with them all the time. I mean, I carry my iPhone with me everywhere I go. So if it recorded my health data, this is going to be a really wonderful way to create that connectivity.
Yeah, so I hope just before this panel discussion, we had awards given out to startups, and we have a great startup culture here in India. I hope some of those people are listening to this conversation and get some ideas about these technologies and particularly their application to clinical trials. Inge, what do you think of the other area that I touched upon, decentralized clinical trials? Are you seeing more of those across the world? Where are we with them, and what do you see for the future?
Yeah, I cannot speak, to be honest, on new drug discovery because that is not my area of expertise. So far in vaccines, we have not, right? So this is because it's quite different. It's prophylactic and patients are followed up. Indeed, I completely agree with what's been said on using the apps on how to get to their next visit or get an alert when they get to the hospital and so forth on the health monitoring. But in terms of implementing decentralized trials, I don't think we have. It will be a slow process for vaccines. I think there are other areas in medicine where this would be more applicable and where it actually can be very beneficial. But for vaccines, I have not seen it so far. No.
Okay, great. So I think we've run out of the time that was allocated to us, and we are actually just on time. So. Thank you all so much for such an interesting round of discussions. I hope you have a great day ahead. Thank you so much, and I will hand over to the organizers for the last comment.
Thank you. Thank you so much, Mr. Mukherjee. And thank you to all our panelists. Thank you for that insightful discussion. We thank you on behalf of OPPI.